Thank you very much, Kati, for inviting me. It's a pleasure to be here. I've never been to Bloomington before, so it's great to see all the amazing work that people are doing and learning. I'll be learning more about that today. So I'm going to tell you about just the concept of mapping the multiscale human, which has come out of this CIFAR program that Kati mentioned that we co-direct. And so CIFAR is a funding agency that brings people together to think about specific topics, and it just funds workshops to help people connect. And some of the programs have been running for many years, like 40 years. So it's an interesting idea of funding idea generation, and we have been funded for discussing the topic of the multiscale human. So um, some of the so the major goals are to understand how the whole human body works across scales, from molecules up to the whole body. And one of the things that we realized in discussions in this working group is that people who are trying to understand the human body have done so in various different fields, and these fields have typically been quite siloed. So for instance, molecular and cell biology have really focused on the scale of the molecules to the cells, and physiology and medicine has typically focused on anatomy, gross anatomy level and higher, and and frequently they don't talk to each other very much. Um, However, they're all interested in understanding the body. So one of the things that's changed recently that might allow us to change this is that single cell and spatial transcriptomics or genomics technology has uh, enabled us to map a lot of multicellular and tissue level information, which bridges the spatial scales between cellular and tissue or organ level scales. And so maybe we can think about developing a unified field that considers how the whole body works across scales, or can we, is an interesting question. So one of the ideas for how to do this that came out of these discussions is to think about the genome as the ultimate generative model. So the joke part of it is that the ultimate generative model because the genome generates people and people generate AI. But the more serious perspective is that um, the we can think about the genome as basically learning through evolution parameters for how to generate a biological system. And for the human genome, it can generate humans. There's also environmental influences and that come into play, but a lot of the information to generate the human system, all the systems in the human body are encoded in the genome. And so maybe we can think about it as a generative system. And this figure here is an example of a generative system that's a transformer that's used in like ChatGPT type of AI applications. This is pretty powerful. There's kind of two implications that come from this thinking. So one is that if the genome contains all the information to generate all of the systems in the body, then anything that we measure about those systems at any scale, whether it's single cell scale or whole body scale, or even all of the literature that describes any of those systems, which is also information about those systems, should be correlated somehow. And you might be able to understand automatically the correlation between these things and automatically integrate them using machine learning potentially. And the other idea is that, especially in the area of machine learning / AI, the current generation of methods is very powerful for automatically learning or modeling data. In particular, the idea of self-supervision allows AI systems to automatically learn without having any kind of training data presented to them. So in the past, we had to spend a lot of time curating positive and negative examples, and now we can just feed data into these systems and they automatically learn how to model them. So for instance, with ChatGPT, they just gave it tons of text, and you can define tasks for it to learn how to complete, like remove some words and predict the word that should have been there and then compare it to the held out word, or predict the next word. And just a simple little task allowed ChatGPT to these large language models to learn the English language and have a conversation with people. There's a little bit more to it than that, but that self-supervision type of technology is very powerful. And so maybe we can use it to automatically learn how the body works or how biological system works. So is machine learning a good approach for understanding the body? We argue that it is, because fundamentally the process that generates these systems is evolution, and evolution fundamentally also has two processes, copying and mutation. It also memorizes and has a selection system, but in terms of copying, that generates redundancy. So most things that we measure in biology are very redundant, like all cells are very similar to each other. And mutation generates variation. And this is great for machine learning because redundancy helps with pattern recognition. And variation, if we didn't have variation, we couldn't do correlation, causation, we couldn't do regression. So that's required. And variation helps link data measurements, for instance. And we do need to keep in mind that we need to think about, we need to understand mechanism. We don't want to have the machine learning system just be a black box, because that's not very helpful for humans for multiple reasons. One reason is that rare events will likely require mechanism-based interpretation. So if you want to if you want to predict the consequence of a mutation in the population, somebody has a mutation, and there's one in a billion people who have that mutation. Well, we need to understand all the mechanisms that that mutation will affect to predict what its effect might be. We can't just give it to a black box that has never seen that mutation before. We might not be able to easily do that. You could argue some philosophy around that, but regardless, I think we want to have a requirement be that we maintain mechanistic understanding. And we also need to think about multi-scale systems, which are typically not thought about in machine learning. I'm going to skip this slide, but if people are interested in talking about the theory of how information might be encoded in the genome, I can come back to it. Genetics is going to be important because that provides us a lot of multi-scale information, because we have a lot of information about how mutations affect different systems at different scales. Like a mutation can affect a protein, a complex, a pathway, all the way up to a whole body to cause a disease. And there's large biobanks that contain much of this information that we can use. And so we'd like to combine that information together. So how would we approach this problem? So one idea, so there would be two parts, mapping and modeling, or structure and function. So mapping the human body is there are many projects like the ones Kati is leading and heavily participating in. I also work a lot with the Human Cell Atlas project to collect information about a lot of cellular and tissue level information. Over time, of course, we'll need to collect whole organs and whole body systems. But the more information we can collect here, the better across the whole system. And then on the understanding side, we might test our understanding of whatever generative models so potentially a generative model could either automatically learn itself, or we can create one. And we need to figure out how to test that. So we'd like it to be mechanistic, or for us to at least maintain mechanistic understanding. And we can test its predictions. For instance, if it's good at predicting response to perturbation, that might be one nice way of testing whether these models are generative models are capturing the system effectively. But maybe a more difficult one, challenging one, would be predicting emergent properties of the system. So if you give the model information at one scale, can it predict patterns that emerge at a higher level that are dependent on it, that we might not even understand or be able to predict very easily, but we might be surprised by, but are important in terms of the relationships between the scales. And if we can do this, there would be many useful applications also in medicine. So I'm going to take you through, so that was just an introduction of some of the ideas that have come out of the discussions at the CIFAR Multiscale Human Program. And I'm going to take you through some projects that we're working on that touch some of these ideas. So the first one is 3D mapping of human tissues at the nanoscale resolution using volumetric electron microscopy. And this is a project with Ian McGilvery, who's a liver transplant surgeon, Sonia McParland, who's a liver genomics and immunologist. Bo is a machine learning expert. May is a neurodevelopmental biologist, but she happens to be an expert in this volumetric electron microscopy imaging technique. And Ronald is a student in my lab, a PhD student who did this work. So the idea with this system, and we're going to focus on the liver, we have a lot of human liver projects, and so a number of the data that I'll be showing you today is about the human liver. But in this case, we can image a volume of the liver, tiny volume, where the pixels are like 1 or 2 or maybe up to 8 nanometers in resolution. And the challenge is to take these kind of grayscale images and annotate them to identify all the structure that is present in them. And I'll just show you a video that explains how this imaging technology works. We basically take a tissue and we embed it in a plastic hard resin, and we do serial sectioning of the tissue. There's multiple different ways of doing that. One of them is with an ultra microtome diamond knife that vibrates, and it shaves the tissue at 50 nanometer slices, and we can image each slice after it's sliced. There's various different ideas of how to actually do the slices, but that's the general idea. And ultimately, we get once we put the slices together in a Z-stack, we get this image. And as I said, the challenge is to identify the objects in this image. So the way that volumetric electron microscopy works now, like in May's lab, she studies the C. elegans worm and images the whole animal. It takes them 2,000 hours of time to annotate manually the image. And then they do it again to double check their work. It's extremely time consuming. And they have very little automation in this field, although a number of people are trying to automate that annotation work. Here's a couple of example images of liver tissue. So what's really neat with this electron microscopy images is that it kind of reminds me, at least, that the cell is a complicated machine that has lots of detailed structures. In fact, in this image, it's hard to even see where the boundaries of the cells are. You can hardly see where one cell stops and starts, and when one stops and one starts. And if you zoom in, you'll see that the cell membranes are actually like really tightly connected into each other, which is very interesting. So we approach this problem using machine learning, and I'll just tell you about a project that is working towards solving this problem of automatically annotating these images. So a lot of our projects, we actually have to provide supervised, we approach the problem with supervised machine learning, so we provide examples, and it then learns we can train a machine learning system to annotate different parts of the cells and whole cells. In this case, we used an unsupervised method, which means we don't need to provide it anything other than the images to automatically identify these objects. So the project is called Master, and it's a self-supervised masked autoencoder, and I'll tell you how that works, that takes the set of 2D slices and automatically identifies objects in each slice, and then you can put those slices together to get 3D volumes. So the way that this works is we take these 2D images and we split the images up into patches, and these square patches, and we mask some of the patches. So we remove some of the patches, and then we convert that set of patches into a vector with a vision transformer encoder, and um, and then we feed that into an autoencoder. And the way the autoencoder works is it takes information as input and then tries to learn how to recreate that information as output. And when you get the output that's decoded from the internal representation that it's learning, you check how close it is to the input. And then the learning algorithm automatically tries to learn an encoding or a high dimensional representation of the input data such that it can decode it accurately. And so over many iterations, it learns a good encoding that helps it make an accurate, you know, encoder to decoder loop. So you can think about it as learning the essence of the data that's coming in. And then once you've learned that essence, you can do things with it. Um, so, so what we do is we feed the images, the masked images, and sorry, the masking here. So normally in autoencoder, you don't mask the input data, but if you mask it, it makes the problem more challenging. And the idea is that the autoencoder will learn a better representation because it's, if you challenge machine learning systems, then they usually have to learn, they have to work harder to learn a more generalized representation, and so that's usually a good thing. Um, so, so we take our images, we encode them into a number of dimensions that we choose, and that's called the embedding. So we embed the images into this high dimensional space, and then we can visualize that space as a UMAP, for instance. So I'm just going to go here. Like this is an example of all of these dots here are image patches, and they're organized automatically by the autoencoder to group together into similar. It's somehow defines similarities between them. And so once we have that, we can cluster that space. So the colors here represent different clusters, and it turns out those are the objects in the, or are object types in the original image. So once we cluster those, we can color the original image to basically be colored a different color for every cluster. And then Ronald figured out an interesting way of kind of doing increasing the resolution than just the patch resolution. I won't go into that, but it's described in the paper. And this just shows you some examples of how good the autoencoder is. If we take the original data and we know what these structures are, mitochondria, nucleus, and we mask 25% of the image, the autoencoder is able to learn this image, is able to decode this image from the internal representation that it learns, and that's pretty similar to the original. If we do 50% masking, it's still able to learn the major structures, although you can see it getting a little bit fuzzier. And even if we mask 85% of the images, it's able to learn the major structures. So the autoencoder does a pretty good job of learning from given enough information. Learning an internal representation. So a few hundred, yeah. So so the question was how many total images did it get to see? It's a few hundred images. So so I talked about this. This is automatic segmentation by a clustering. I explained that, and we did these. We used k-means clustering, nothing fancy. And we could probably get more information out of this than we do. But for instance, it looks like there might be different types of mitochondria, and so we're in the process of thinking about that and going into that in more detail. But right now, for this paper, we just used a simple clustering method to identify the objects. So here's the original image as an example. Here's the reference segmentation that was manually annotated into these classes and these categories. And this is what Meister automatically identifies. It's not perfect. So you see that some of the things are missed. There's a little bit of fuzziness at the boundaries, but it does a pretty good job, given that we don't have to give it any training. The only training data we give it is the images themselves. And we are now working to improve this and combine it with different, there's different strategies that we can do to improve it. But I think as a proof of concept, it shows that self-supervised machine learning can do a good job of understanding biological representations that we can use for practical applications like automatically annotating images and 3D volumes. The next project I'm going to talk about is also using machine learning, but more related to comparing different types of information. Again, this is a project that Ronald helped lead, but it was actually done by a summer student, Quan Pan. Also, Bo is helping with this project. And this is also published in a computer science conference. So the idea with this project is we wanted to take spatial transcriptomics data where we have an image of a tissue that is, in this case, it's a slice of liver tissue, human liver tissue, and it's stained with H&E staining, which is a standard type of stain that stains different cellular structures. And that image in spatial transcriptomics is typically used just for quality control, but there's a lot of interesting information here. On the other hand, we measure the location that genes are expressed in, which is the spatial transcriptome. And we wanted to understand if we could automatically figure out the relationship between these. So Ronald developed this LEAP method that tries to predict the spatial transcriptome from the image, or vice versa. Okay, so how does this work? So what we do is we use machine learning to learn a joint embedding of the image and the transcriptome. And that's for people who are familiar with those words, but similarly to what I showed you before, we take the image and we split it up into patches, and we encode that image using an off-the-shelf image encoder. It was, I think, ResNet in this case. And that generates it kind of maps the images into a space, an embedding, embeds it in a space. And we do something similar for the transcriptome. So in this case, we're using Visium, 10x Genomics Visium spatial transcriptomics technology. And Visium measures transcriptomes as a series of spots, spatial transcriptomes as a series of spots. So each spot is basically measuring the gene expression levels of the tissue above it. So you can imagine like a little square here of this image. We have a corresponding dot that measures a transcriptome. So it's a vector of genes and their expression levels that is averaged over all the cells in that little patch. And we encode that information, again, embedded in a space. And then we apply contrastive learning to automatically learn a joint embedding that maintains the similarity of the images to each other in its image space and the similarity of the transcriptome measurements in its space in such a way that the joint embedding keeps those, if two patches are similar in the joint in the image space and two corresponding transcriptomes are similar in its space, in joint space they should also be similar. And once you learn that, you can translate from one to the other. So we can take an image, we can convert into patches, and we can read out a transcriptome from each of those patches predicted from the system. And here's an example, a good example of how it works. So this is an original overlay of a specific gene, the CYP3A4 gene, and you can see its expression level across space, where yellow is high. And just given the image, BLEEP is able to predict this pattern, and that correlation is around 74%. So that's very good. Most of the genes, so that's our best example. There are other genes that have high correlation, but on average, the correlation is only just around 20%. That beat out other methods that existed at the time, but there's still a lot of room for improvement. This is now a bit of a hot field. There's a lot of papers coming out, and they're driving the prediction performance up. This project also only used 4 Visium samples just with human liver. It doesn't readily translate out to other tissues. So we're working on next generation systems that use a lot more data and develop a better, more generalized representation of this image transcriptome relationship. So this project illustrates that idea that I mentioned in the beginning, that we can use self-supervised learning to, or versions of it, to automatically learn relationships between data measurements that we're making. So if we can make measurements, and one of the things that would be interesting to do in the future, just see if it's possible to do that across scales. Right now, I don't know if anyone's tried that. Just checking my amount of time I have. I'm going to skip through this, but I just wanted to highlight an interesting use of ChatGPT. Ronald wanted to collect more data, and he didn't want to sift through the database and annotate manually all of the labels of the types of tissues and diseases that they represented. So he used GPT and was able to automate curation of the geodatabase to find all the spatial transcriptomics in a couple of hours one afternoon. Fortunately, he asked another student in my lab who works on text mining to help him, but just with some prompt engineering, we were able to collect all the metadata from the gene expression omnibus and curate it using, for people that are familiar with this, there's 2 techniques that we used from prompt engineering, just general prompts and also reflection, where you ask the system afterwards to tell you why it chose the answer and how confident it is. I'm not going to go through the details, but it's a useful maybe example of how it's an interesting example of how ChatGPT technology can be useful for automating curation. I'm going to skip this as well, this slide. Okay, so the next topic I'm going to talk about is a project that Maria, who's a research associate in my lab, is helping coordinate with Nozomu Yachi and Nika Shakiba, who are bioengineering professors at the University of British Columbia in Vancouver, called the Virtual Human Development Project. And this is an open project that anyone who is interested in modeling human development can join, and they have been able to connect with dozens of labs around the world. I think James Glazer's lab is participating, and it's open for anyone who's interested. But basically, they want to develop methods to simulate human development and link it to experiment and have a really good conversation and cycles of development, iterative development that include experimentalists and modelers working together to develop good models of human development. So I'm going to talk about 3 projects that are related to human development in general, or stem cell biology. 2 of them are in machine learning, and one of them is mathematical modeling. So the machine learning ones I'll talk about first. This is work done by Murshad Sadria, who's a physics PhD student at the University of Waterloo near Toronto, and who's kind of visiting my lab. He's been very interested in understanding how cells make decisions about as they're growing, about which cell they're going to become. So stem cells start out as just one type of cell, the pluripotent stem cell, that is able to generate all the other cells in the human body. And it does that through a series of steps that generate different cell types, and those change and progressively get to be mature, the final mature product. an important question that we still don't understand is how those cells decide, like, how to change and differentiate. So what Mershad did is he decided to take single-cell transcriptomics data of developing systems, like developing tissue, like developing blood system, for instance, and see if he could figure out ways to identify cell fate regulators in that data. So what he came up with is to model this data using an autoencoder. I should have mentioned autoencoders, kind of very similar to principal component analysis. It just has the added benefit of being able to learn nonlinear relationships. So it's in this case, it's we feed in all the transcriptomics data and we learn an internal representation. He added one for the aficionados. He added one extra bit of this that is tries to use an additional classifier in the loss function that tries to predict, as this system is learning, whether the internal representation is useful for a task, in this case, classifying which cell type was present in this original data. And that, some people have suggested, allows you to learn a better representation than if you didn't have that constraint, and that additional constraint. So once we learn the internal representation of this data, then we can knock out these nodes, these internal nodes that represent parts of the internal representation. And we do that, let's say we knock out one of these nodes, we call it a in silico perturbation. We decode the result, and the decoded result is a gene expression matrix, like genes by cells. And once we have that decoded gene by cell matrix, that's the predicted result of the in silico perturbation, we can apply a standard cell type classifier to it, and we can see what cells are there in the result. And so, oops, I forgot to include a slide here that shows an example of this, but the perturbation, as we modify the parameters in the middle here by setting, the values here to 0 or some other value, we multiply these numbers by a value like 0.5 or 10. We can see cell type classification change in the output. And we do this for all the different nodes, and we generate a database of results. So then we can look at that database of results, and we can say, if we're interested in converting one cell type to another, when I perturb this node, I get gene expression pattern that has converted this one cell type A to cell type B. And so it's kind of like a in silico CRISPR screen to identify cell fate regulate. Sorry, I forgot to mention one thing, which is that once we do that, we're able to figure out what genes or gene expression pattern is kind of captured by these nodes that were internal representation nodes that we're perturbing, and we can generate a list of genes that are associated with that node, and we can look through that list to identify cell fate regulators like transcription factors. So recently, with Miriam Fayez, who is a stem cell biologist in the University of Toronto, we were able to test this system experimentally. So she is interested in converting astrocytes, converting different types of cells in the brain to each other for regenerative medicine purposes. So in this one project, she's interested in converting astrocytes to oligodendrocytes. And the way that the conversion works is that they introduce a gene, like a transcription factor, into the astrocytes, and then it activates programs that convert the astrocyte into oligodendrocytes. And if you did that in a human brain in the future, you may be able to create cells that are not getting created during an injury condition, and that converting or adding new cells of a particular type might help repair the damage. So that's why she's interested in it. So we applied fate code to her data, and fate code predicted SOX10 as the top transcription factor responsible for driving cells that towards one of the clusters in her data that represent oligodendrocyte lineage cells. And so in her data, there's a whole bunch of different clusters that she identified. It was actually, this is the data here. I should have put it on the slide as well to show you. But so these different cell types are ones that are in the data. And then when we apply fate code, we can find a condition that converts. So this is the target cell type that they're interested in. And we found a perturbation that predicts a gene expression matrix where there's more of these cells that exist. And there are fewer cells of some of these other cell types. So the blue region is sort of measures the amount of cells that are kind of being removed from this cell type and this cell type and this cell type. And this orange part here shows that there's more of these cells being produced given a perturbation. So fate code predicts that SOX10 is important at creating more of these oligodendrocyte lineage cells, and they tested that by expressing SOX10, and it does induce increased growth of oligodendrocytes in the mouse. So we hope that fate code can be useful for experimentalists to identify candidates to test in these types of experiments. Oh, here's the image, the slide that I was going to show before that I forgot. I'll just go through it quickly. The perturbation values here show all the different experiments that we're doing. And here's the cell types that are generated. And you can see that one of the perturbations at some point generates much more of these yellow or greenish cells and fewer of the red cells. And then we were able to predict important genes that are associated with this. And you can see the ones that are changed and unchanged by this perturbation, all in silico. Okay, so the second project, also by Murshad, that follows this this task is a little bit more takes a little bit more information and considers a little bit more information. So we call it deep lineage. So one of the things that you can do when you are growing cells is you can put in markers that or barcodes, let's say DNA barcodes is a kind of marker, and you could trace the lineages. So as the cells divide, you can trace their ancestors or their progenitors. So and people have come up with all sorts of interesting ways of doing this that allow you to read out the history of the cell division events for even a whole animal. And this is called lineage tracing. And so this information exists in single cell experiments. So it's basically a modification to single cell transcriptomics data that allows you to kind of insert a molecular recording device and then read out the trace afterwards and then infer back the history once you've looked at the trace. And so Murshad has developed a system that is a modification of FateNet that considers this additional data and also time series data. So we have the cell, the time point that it's at, the clone that it's part of, or that it might generate progenitors that we can map and link to each other with these lineage tracing, and the expression values. And we learn that whole thing, and we use a time series-based deep learning model, recurrent neural network, to understand the relationship between the time points. And so what this does is it models a time series, and once we've modeled the time series, we can predict the expression that will happen at any time, including in the future that we haven't even measured. So the way we tested this is we have an experiment that had measured gene expression data on day 2, 4, and 6 from a blood, blood development study. And we hold out or hide one of the days and train the system So we train the system on the whole data set, and then we give it day 2 and 6 data, and we say, predict day 4 data. And we've held out day 4 data. And the prediction is very good. We get high prediction accuracy at predicting any held out time point. And even at the gene expression level, just with a set of random genes, we get quite good representation, prediction of what the gene expression should be at that day compared to the real day, the real data. And so this allows us to kind of do forecasting of, we would predict that if you, and we identify genes that are important. So you could combine this with the perturbation experiments to perturb early stages. So either identify factors at early stages that predict later stages, or perturb the system so that you can change it in silico, and that might help you in engineering applications where you're trying to engineer the generation of certain types of cells in your tissue. Okay, so I'm going to skip to mathematical modeling now, and this is work by Maria Abuchakra and my group, who's been thinking about how cells grow from a stem cell. So we have created a cell development simulation model in 2D and 3D, and we have all sorts of parameters that we've been playing with. You can generate like a 3D model that looks like this of cells growing, and we can change parameters and see the result. For this project, I'm just going to talk about a 2D version of it, or not even 2D, but even non-spatial version of it, where we just call it a digital cell model. So in this project, we want to understand how cells are, again, making decisions about how they generate tissues or lineages, cell lineages. And we start off with a single cell, and we have an agent-based model that has a few different components. So it has a genome. So each cell is represented as an agent, and each agent has a genome with a set of genes on it, and the genes can express, and there's a gene regulatory network that's incorporated, and the cell division event can occur, and at cell division there's a random so in the beginning there's a cell, it generates genes from its genome, and then there's a cell division event that occurs that randomly distributes the transcripts to the children cells, and we can just keep on doing that as many cell divisions as we want. We also have a parameter for the duration, the cell cycle duration, which is how long it takes for transcription to occur. And that's very important because one of the things we learned about in this project is that when cells divide, they don't always divide in with the exact same time as each other. So there's a lot of variability in how quickly the cell cycle runs. In early development, it's really fast, and it can be as little as a minute or two, where in later time points, you might have cell divisions that are taking 40 hours. So there's huge variability in the length of the cell cycle. And this is important for gene expression because when genes are expressed, transcription occurs as a linear process that has a particular rate, and it takes time. If you have a very long gene and a short cell cycle, you won't have time to finish transcribing that message before the cell divides. And when the cell divides, all of that transcription gets interfered with, and you basically lose all of those transcripts that haven't been finished at that point. So we've been investigating how that combination of parameters, the length of the gene, and the cell cycle duration can be used to define or control gene expression patterns. And we had a paper in eLife in 2021 that had a model that talked about this model that didn't have gene regulatory networks. It was just a simpler model, but it showed that there's a trade-off between when you're developing a tissue or growing a set of cells, like a cell lineage, you can trade off how many cells you generate for the diversity. So the way that works is that if you're cycling very fast, you can generate lots of cells quickly because you're just making lots of cell division. So it's like major production of cells. But that reduces the amount of your genome that you can express. So there's a bunch of long genes that can't be expressed when you're producing many cells. And so as you slow down the cell cycle, you reduce the number of cells that you can make, but you add more genes, and that increases the diversity of your system. And you and so we showed that you have this trade-off between kind of production of many cells and diversity. And by tuning that, we think that that's one of the mechanisms that operates in development to set up the structures of multicellular systems. In this work, we've added gene regulatory networks, and when we run these simulations, we can get the gene expression values over time of our theoretical in silico genes, and we can each cell is generated with a transcriptome, so it's like a single cell transcriptomics experiment. So we can identify the states of the cells, and so what we get out of the simulation is a population of cell states that have different proportions. And the cell states are defined based on the genes that are expressed in the cells, just like transcription factors define frequently or other gene expression markers define cell types. This is a little bit more information about the parameters that we have that I won't go into. Okay, so this is unpublished work, but we're hoping to submit it soon. So we did a lot of simulations. We did like 10 billion cells. We did huge numbers of simulations where we changed all these parameters as much as we could. And when we looked at the results, we found that there were 3 core regulatory patterns that we could identify. We can kind of group all of the results into 3 core ideas. So one is a toggle switch. So depending on the length of the gene or the copy number or depending on the gene length or the copy number or the regulatory circuit that is present, there's a number of different combinations of parameters that can lead to the same pattern. There's one type of pattern that exists where you just choose which gene is expressed high and which gene is expressed low. So you can choose to switch the gene to be high or low. It's like one's high and one's low, and you can choose which one is high. In this experiment, we did only 2 genes because we did so many millions of simulations that we couldn't do more complicated systems. So this is a very theoretical system, but we hope it illustrates some fundamental concepts in the topic. Okay, so one idea is a toggle switch. The other pattern we saw was a time-shifted toggle switch. So this allows you to set up the system. You can choose the parameters to generate gene expression profiles where you choose when the switch occurs. So in this toggle switch, it's either on or off. In this time-shifted toggle switch, you can set the parameters to tune when you see a shift. So you can see, for instance, here the switch is early, and then here it's kind of later. Here's an example where it might be even later. In essence, we can choose these parameters to choose where that switch occurs. And the third pattern is a controllable switch where the system can actively control the dynamics of when that switch occurs. So in these first ones, these parameters are set. Like once you set the length of the gene, that's set by evolution. It doesn't change in the development of the organism. Same thing with a copy number, and probably the gene regulatory network is kind of defined as a starting point. You can change the dynamic, you can turn genes on and off once the system gets going, but it's kind of set. Cell cycle duration can be regulated by the cell, and I guess you could say that gene regulatory networks can as well. But in this case, we found that cell cycle duration was a very important parameter that allowed you to tune when this switch between A and B would happen, and even make more complicated patterns where you switch, A is high, and then B is high, and then A is high again. So ultimately, we organized all of this information into something we're calling a cell lineage generation code, where we have all of our parameters and we kind of understand how they influence pattern generation in the system. So each of these little pie charts here is a set of cells that is the result of the simulation, and we can consider it a pattern that's being generated. And the and we can basically, this code allows us to set the parameters to generate patterns that we'd like to see. So these blue lines here represent, well, each kind of developmental time where we shift from a fast cell cycle to a slow cell cycle, which is something that occurs in early development, as I mentioned. So this regime here is when genes are the same length. This regime here is when the genes are different lengths. And here are the different parameters that we can choose to generate different patterns. So we hope that this organization of all this, these parameters into this kind of single figure can help in the future for engineering well, one, to understand how patterns might be generated in the real system, but also in the future to understand how to change these parameters to generate different patterns. I'm going to skip this. This is work we did to compare it to a real system, but we did try more complicated gene regulatory networks and compare it to real data and found some interesting similarities. But ultimately, it's very, very theoretical, but it illustrates, coming back to what I mentioned, that we have a generative system. In this case, it's an agent-based model that generates patterns, and we're trying to understand how those what parameters influence the patterns that are generated. And hopefully that helps us understand how the real system is working. Okay, so that's all I wanted to talk about today. And I acknowledged Maria, who did the recent work, Ronald, who did the BLEEP and Maester, Mershad, who did the other cell-fate prediction, and the other people in my lab have contributed in the background into other projects we're working on. And this is current and past people in my lab and our funders. Thanks.